It's our pleasure to have Professor Guhan Jayaraman with us now. He has joined 5 minutes ago, and we had some informal chat. And I welcome for all the participants for this session. And on behalf of Madurai Kamaraj University, we are really proud and pleasure in inviting Professor Guhan Jayaraman, Professor in Head, Department of Biotechnology, IIT Madras. He basically, Dr. Guhan Jayaraman is a BTech graduate from Banaras Hindu University, Varanasi, and got his PhD in the year 1993 from Rensselaer Polytechnic Institute, New York, USA, in the year 1993. And professor is there working in the IIT since 2009 as in the Department of Biotechnology. He has published more than 70 papers in highly reputed international journals and having a H-index of 20. And he has operated several research projects, government-funded research projects. Some examples are DBT-funded and BIRAC-funded projects in the related area, what he is going to present today. And he is a faculty in charge of the BIRAC-funded Bio Incubator Facility at IIT Madras. That is really a prestigious capacity he is holding. IIT Madras Research Park is having that a faculty in charge of BIRAC-funded incubator facility. And he is also instrumental in establishing the BIRAC-funded yearly translational accelerator to facilitate technology transfer from academic institutions to industry. That's really wonderful and really wanted for this country. Dr. Jayaraman has undertaken several industrial consultancy projects. That is how the IIT Madras is ranking top in all the rankings because institutional collaboration. His area in industry collaboration is recombinant protein production to biofuels and nutraceuticals. And he is also one of the joint recipient of the BIRAC Innovator Award in the year 2019 and also mentored the IIT Madras team, which won the iGEM 2008 awards in foundational advances and best engineered biobrick categories. So we are having a very highly suitable dynamic researcher for the theme of our AICTE sponsored ATAL-FDP program on synthetic biology. Welcome you, sir, on behalf of the authorities of Madurai Kamaraj University and on behalf of the organizing committee and on behalf of all the participants. Sir, now the podium is yours. You please take your session, sir. Thank you. Thank you.
Thank you. Thank you very much for this kind introduction and giving me the opportunity to give a talk in this program on synthetic biology. As I understand, there are more than 200 participants from all over the country, nearly about 80, 90 universities. And one disadvantage of giving perhaps the last talk in a workshop is usually that you tend to repeat some of the ideas and concepts which have been spoken in the last 3-4 days. And I was just asking whether I'm likely to repeat, and I wanted to know the titles. But from the looks of it, I can relax because not many would have talked about metabolic engineering. So my research essentially is in the area of metabolic engineering, and for which I'm trying to apply synthetic biology tools, essentially, as you can see in the subtitle, for control of gene expression and metabolic fluxes. And for the last few years, I've been focusing on a very specific product called hyaluronic acid, which is a biopolymer, which has a lot of biomedical applications. I'll talk about that later. But of late, I've also diversified into other categories of products and applications in metabolic engineering. So essentially, I will start this talk by giving a brief overview, an intro to metabolic engineering, in case many of you are not familiar about that field. Then I will talk about what are the synthetic biology tools and approaches and how they are applied to metabolic engineering. And finally, I will show one example of a synthetic biology tool which we are applying for our own research on hyaluronic acid production. So essentially, the focus of metabolic engineering is to engineer microbes that can be used for cost-effective production of fuels, chemicals, and biopharmaceuticals, right? And over the last 2 decades, 15 years or so, there have been a variety of products, as you can see in the slide, a variety of products which have been evolved. Many of them, of course, are at the research stage and pilot scale stage, but there are many which have found are starting to find commercial applications, and these vary from products which are bulk chemicals and biofuels to those which are applications in polymers and bioplastics, to surfactants, to fuels I just said, and also in the area of nutraceuticals and drugs and so on and so forth. So traditionally, you can say, hey, well, metabolic engineering seems to be covering what traditional industrial biotechnology does. So how is it any different from traditional bioprocess engineering? Well, there are some vital differences. A, you can always produce novel products by engineering pathways from other organisms, or even by engineering many of the enzymes, which cannot be produced by any other means. So a lot of this is perhaps the holy grail of synthetic biology, and there are few applications on this. Artemisinin is one great example which has been touted about in the last few years. But there are not too many products like that. But essentially, it is to engineer microbes to do better than what native organisms can do. And this could be in the use of raw materials, for example, lignocellulosic biomass, to replace fossil fuels. And you can say this is already being done, right? And there are a lot of applications. But other than biofuel, we have to remember that there are a lot of carbon-based petrochemicals, and the only substitute for petroleum, the only carbon substitute is biomass, right? There are many non-carbon alternatives to petroleum, which are energy sources, which be it nuclear or solar or wind energy and so on. But the only carbon source is biomass. And so that's extremely important, and the future applications of metabolic engineering will see organisms being engineered to produce many, many different kinds of petrochemicals, and this is extremely important in the context of climate change. If you think that COVID-19 has really been problematic this year and has created a huge recessionary trend, which tends to dwarf even what the problems of COVID-19 are, climate change is multifold, of greater magnitude than what the whole havoc wreaked by COVID-19. So just think about that. Okay, so it is very, very important that if you are going to go to a green economy, be it for the use of biomass as a carbon source to produce a variety of chemicals, or to have green processes to replace chemical processes such that they are more environmentally friendly and so on. The bioeconomy and what we call as biorefineries will play a very, very critical role in the future economy. And in this, metabolic engineering itself has a very critical role. So other than that, we can also use metabolic engineering as an alternative, especially metabolic engineering of bacteria, yeast, and microorganisms. I think there is a background noise. I would request everyone to mute. I'm getting some background noise. Thank you. So it can also be used as an alternative to extraction. Sorry. Yeah, extraction of chemicals from plants or animal sources. For example, the artemisinin example I talked about, which comes from plants, now can be produced by bacteria. There are anti-cancer drugs which used to be extracted from the bark of plants, now which can be produced by bacteria. So it can serve as a more economical alternative and a better microbial alternative. And in many cases, we can perhaps even use metabolic engineering to produce products by replacing native hosts, which may be pathogenic, right? So we could have what we call a safer host for production. And the last part of my lecture is going to focus on this kind of topic on hyaluronic acid production by one organism which we use in our lab, Lactococcus lactis, which is a food grade organism, right? And we have used that to produce hyaluronic acid instead of the native producer, which is Streptococcus zoopidemicus, which is a pathogenic organism. So it can have, as you can see, there are many, many different reasons or many different applications of metabolic engineering. So what are the ways we try to implement? What is the objective? These are the applications. As I said, for the bulk chemicals, what we call as low value, high volume products. This could be biofuels, or it could be bulk chemicals, like lactic acid or semi-bulk chemicals like amino acids, not of course bulk to the same degree as biofuels, but nevertheless fairly high volume. So in all these cases, the important criteria are what are known as the TRI criteria: titer, rate, and yield. And this is true for all bioprocesses, right? And metabolic engineering tools only seek to enhance the parameters in these three parameters of the TRI criteria. One is to enhance product concentrations, okay? And if you have much higher product concentrations in your bioreactor, that tends to lower downstream processing costs. And in case many of you are not familiar, downstream processing costs or purification costs can be as much as 50 to 90% of overall process costs. Okay, so it is therefore very, very important to enhance titers in any bioprocess. And sometimes there are many products which can inhibit the growth of the microorganism, and therefore one cannot go beyond a particular concentration. Okay, and there are ways to engineer organisms to enhance the overall titer. Rate or productivity, which is the amount of product which we produce per unit volume, per unit time, can also be enhanced by increasing the flux in the pathway which is used for product formation, and enhancing the kinetics of individual steps. So that is extremely important. Okay, and there are ways of doing it in metabolic engineering, which normal organisms are, might find it difficult to do. Also, the third criteria is yield, because that's an important economic criteria. If you're using any raw material, and especially if that raw material is expensive, you want as much of product being produced from that raw material and not allowing that raw material to be diverted to byproducts. So essentially, this yield talks about the amount of product formed per unit amount of substrate that's taken up. And so one can enhance the yield by lowering the byproduct formation, diverting more and more substrate towards product formation. So that also involves altering of many of the metabolic pathways within an organism. So one can enhance the product yield. So these three criteria, remember, are extremely important, And this is especially true for what we call as the bulk products, the low value, high volume products. Also, we would want to perhaps use a range of substrates, right? Especially if many of these substrates are cheap and they are waste substrates. And often the natural organisms do not grow on all kinds of substrates. They may have a preferred substrate. They may, have very tightly regulated metabolic pathways, and catabolite repression might not allow any one substrate to be used, and so on. So essentially, one goal of metabolic engineering is also to make the organism highly flexible by adapting to a range of substrates, by adapting to what we call as industrial media, etc. So be it something like lignocellulosics, right, which could have three different components. It could have cellulose, which lead to glucose. It could have hemicellulose, which lead to pentosis. It could have lignin, which leads to different kinds of monomers. And a bacteria may not take up all these different substrates. So you might want to engineer bacteria to at least take up maybe celluloses or glucose and pentoses simultaneously. Or you might want to engineer organism to adapt to use a lot of glycerol from the biodiesel industry, or carbon dioxide, which anyway you do with cyanobacteria and so on, or whey from dairy industry, or natural gas, which is abundantly present, right? And that's also a greenhouse gas. So these are the challenges. And how do you go about engineering organisms to do that is a big challenge. And as I said, the other problem which also comes is that many of the natural organisms may not tolerate either a high concentration of substrate or a high concentration of product. If it doesn't tolerate a high concentration of product, so the titer value goes down. So, or it may not tolerate certain toxic components in the media. For example, if you hydrolyze lignocellulosics, you could have a lot of furfural, which may inhibit growth, or it may not tolerate high product concentrations. So how do you go about engineering the cell so that it can tolerate all these kinds of stress, right? How do you engineer the stress response? Or in some cases, the cells may grow in high cell density where the oxygen limitation may be a problem, right? So can you engineer the cell to adapt to low oxygen environments, which is present in high cell density cultures? So there are various ways one can go about engineering cell. It is not just metabolic pathways. So all these are just a few examples. So all these could be the objectives that we have for engineering an organism to give you the different kinds of applications I just talked about in the previous slide. Then for the other category of products, there are other products which may be low volume, in which case the titer is not very important, or even rate or yield. All these are not important, and these are usually products like drugs or specialty chemicals, etc. Right? We have always seen recombinant therapeutic proteins. The cost of producing that is only a fraction of the product cost, because a lot of the costs are taken up by clinical trials and so on and so forth. So for very high value products, the objectives may be very different. It may be finding a suitable organism which is generally regarded as safe. I just talked about Lactococcus lactis, right? Replacing maybe a pathogenic organism. So even though E. coli is a widely used organism in synthetic biology because of issues of endotoxins and others, so sometimes people may not want to use E. coli. So you want to engineer the organism, and that might require a lot of tinkering around a particular organism, and I'll come to that point later. The second, and that is even more important, is that even though these products are low volume, the cost is not so much as the issue as product quality, right? In case of recombinant proteins, it may be something like glycosylation patterns. That may be extremely important. It may be, that you want to eliminate certain kinds of degradation of the product. So if it's a protein, especially therapeutic protein, you may not want protein degradation, or you may not want heterogeneity in the product in terms of charge heterogeneity, etc. You may not want any kind of undesirable product modification. So all these things might also require engineering the organism such that the product quality is maintained. I'll talk later also about one kind of product quality which we are trying to maintain in lab, and that is the molecular weight of this particular polymer that we are talking about. It may be eliminating toxic byproducts, be it endotoxins or other things I just talked about. So all these things are important, and often maintaining product quality may also mean that you want to ensure reproducibility of a particular process, and the engineered strain needs to be robust, right? There must not be that much genomic changes in generation to generation. Maybe you should not generate that much amount of heterogeneity, and I'll talk about the issue of heterogeneity later on. You may not want to have a lot of, let's say, plasmid instability, and you may want, consistent gene expression patterns throughout the growth process and in batch to batch and so on. So this is much more critical. So in these cases, the whole objective of metabolic engineering is very different than the previous slide where I talked about that you want to enhance the productivity or the yield or titer, where the main consideration is cost, right? Economics. Here, economics is not a consideration. So this is again in my later slide I'll talk about this, and in this production of hyaluronic acid by Lactococcus lactis, one of the issues of maintaining product quality, apart from the fact that we want to use a safe organism, the product quality is that this hyaluronic acid, which is a high molecular weight polymer, and the polymer molecular weight varies from hundreds of thousands of Daltons or even tens of thousands of Daltons to millions of Daltons, right? 2 orders of magnitude variation molecular weight, and that quality, right, the molecular weight can impact what kind of product application we seek to take. So I'll come back to this whole issue later. Now, if metabolic engineering can play such an important role in bioprocess engineering, why don't we see widespread application of metabolic engineering in industrial biotechnology? That's a question. I mean, the field is only about 25 years old, right? And you say 25 years old is a generation. Synthetic biology is perhaps lesser, maybe 15 years old, or I would say, depending on which stage you define it, maybe even 10 years old, right? And so we are in that stage of biology where a lot of tools are being defined, and there are so many constraints to metabolic engineering which prevents its widespread application. So we look at some of these. Of course, one of the biggest problems is that we have incomplete understanding of metabolic regulation and the phenotypic response to metabolic manipulation. This, I'll not get into this in detail. Maybe I'll talk about it in a couple of slides. But essentially, apart from a very few number of organisms, right, most organisms are pretty much dark biology, right, where we don't know how the organisms will behave, even though we may know the complete genome sequence. We may not understand the metabolic networks, especially we may not understand regulation. So there are a limited number of organisms which we understand very well, and which can be employed in metabolic engineering. And this is only a small number. And of course, the most well-known, of course, is E. coli, in which case we have extensive knowledge of the physiology and metabolism of E. coli, but we may not have it to that extent, for example, with cyanobacteria, right? You may have wide range of engineering tools, genome engineering tools and plasmids, etc., available for E. coli. You may not have it for other organisms. Okay, so different organisms have different strengths. E. coli may be the most widely used tool. That's because a lot is known. It's historically been worked on, but doesn't mean it has natural advantages. For example, people want to work with things like Pseudomonas, right? Pseudomonas has a lot of advantages which E. coli doesn't have, especially in terms of tolerance to different substrates, and you can use it for a lot of production of aromatic compounds and so on. Bacillus subtilis has some natural advantages in producing a lot of enzymes, right? It has very efficient protein secretion systems. So if you want to produce a lot of industrial enzymes, one would use bacillus or fungi. And in case of fungi, perhaps there are lesser number of tools than in case of E. coli or bacillus. So there are different organisms which have a lot of inherent advantages, but it doesn't mean that all of them can be naturally used. So there are still within these classes, one would say that there are perhaps, dozen to probably 2 dozen organisms which can be widely used. But consider that with the thousands or millions of organisms which can have very varied kind of metabolisms, which can have very varied kind of environmental niche, if only we understood them better, if only they had well-developed genetic manipulation tools, right? Then metabolic engineering could become so much easier. So that's a big problem. All that, of course, takes a lot of time. It takes a lot of money. All of you who are working in biology know how difficult it is to put together all this knowledge, right? And naturally, many of these other organisms that you use, even though you might try to engineer it, it may have much lower values of try as compared to a natural organism. Even today, a natural Saccharomyces cerevisiae may be the best thing to produce biofuel, right? There are other issues in metabolic engineering. The moment you want to engineer an organism, you have to go through a lot of regulatory issues, right? So that's a problem. I'm not even going to get into it. I'm going to talk about technical problems. So people, that's why they prefer natural organisms. And even if you want to go to the regulatory route and metabolic engineering, it's easier to get organisms approved, which are well known, like E. coli or Bacillus or Saccharomyces. If you say that, okay, I want to now go on with Lactococcus lactis or some, well, even Lactococcus lactis is reasonably well known, or something even more abstruse, people will say, well, you have to do many, many more kinds of characterization and come back. And so people start to hesitate. So that's a problem. So all these other things I just talked about are there. Many of the organisms grow on limited range of substrates. But most importantly, what one sees in engineered organisms is that people often very easily engineer the pathway of an organism. Okay, they produce a product in shake flasks, and they say, well, I've engineered, it's producing well. But often, if you scale it up and grow it in bioreactors, maybe a lab scale bioreactor like 2-liter bioreactor, you may get very different results from a shake flask. If you take the conditions in a 2-liter bioreactor and scale it up to a 50-liter bioreactor, or to a 500-liter or a 5,000-liter bioreactor, again, the results are likely to be very different. Okay, because the environmental conditions, there's a lot of heterogeneity, and often there is improper validation and optimization of the engineered strains, be it medium optimization or optimization of process parameters, or it could be codon optimization, etc. So all these things need to happen together, hand in hand. I'll again come to this issue, but remember this, right? It's not just enough to engineer a strain. A strain has to work well in a process environment, right? And that often people don't do in the initial part of a process. They may engineer a lot of things, and then at the end of a process, they'll say that, well, it doesn't work too well. So these are things that people realize late, and then they have to go back to the drawing board. And then there are issues of, as I said, genetic instability and loss of reproducibility of after many generations. So organisms are not very robust. And then there are issues of, heterogeneity in populations. Okay, so I'll come to some of these issues a little later. And of course, the basic issue, why we cannot, of course, apply it to a lot of bulk chemicals, even biofuels, is that the costs are very high. So relative to petroleum-based products, it's still high. So unless there are regulations which say thou shalt not use petrol at all, right? You have to use only ethanol, and that too ethanol from biomass. Okay, and it doesn't matter. You have to still pay 150 rupees per liter, and that's going to cost a lot, and it's going to have an economic impact. People will not use these products, and therefore the transition to a bio-based economy has an economic cost, and that is why countries like US or many other countries don't want to do it, right? Because petrol is cheap and other consumer goods are cheap. So why would you want to, burden the consumer with higher costs? So this is the issue. But it will happen at some point. Petroleum is going to get capped. It's going to become much more expensive. And as I said, even if you don't use it for energy, if you're going solar, other carbon products, you'd have to use biomass. So if these are the constraints, how do you overcome them? What are the tools? I just want to ask one question here. In my own presentation, I see that I get blocked by some of these, bars and so on. I hope you are able to see the slides very clearly. Is there an issue? Is this slide
yes, sir,
being
it's
blocked?
clear. It's very clear. Yeah.
Okay. All right. Okay. So, so how do you overcome these constraints, right? And what are the tools for it? And this is something I'm not going to spend too much time. You would have learned from a lot of other lectures. We'll categorize these as 3 or 4 different classes of tools. There are computational tools which one can use. And then there are 2 or 3 different kinds of experimental tools. There are experimental tools for data generation, what we call as the omics kind of data. You would all be familiar with it, be it transcriptomics or even genomics, right? Or a lot of proteomic data, or a lot of metabolomic data, and so on. So there are infinite number of omics. And if you generate a lot of this data, that leads to a much better understanding of the organisms and the constraints which are there in the metabolic pathways and other kinds of genetic regulation and so on, and how to go about. You can start to have an idea of what the target should be and how to overcome that, right? So the first idea to start with would be to simply get a genome sequence, which is now very cheap. And then if you generate all this data, it can be used for a lot of computational modeling, what we call as the genome scale metabolic model. Okay, so we start with the genome sequence, and you have a reconstruction of the metabolic pathways from the genome sequence, and using annotation of these various genes from comparison with other organisms, you'll know what are the different enzymes and whether a particular pathway is intact or there are parts which are missing, right? And then from a lot of the other data, the transcriptomic data and maybe the fermentation data, the proteomic data, one can say that certain genes are not being expressed, certain metabolic pathways are very tightly regulated, and so on and so forth. So one can have a more curated construction based on the data, right? However, you need complete data, physiological data, and a lot of this omics data to have what we call as a genome scale metabolic model, that we know that where the bottlenecks are what pathways function, etc. And then that you can play around with. Okay, you can say that, okay, this is going to be my growth rate, and you can apply constraints to that. You can say that this is going to be my substrate uptake rate, this is my particular substrate, or if I'm going to mutate genes here in these pathways, how's the flux going to get redistributed, and so on and so forth, right? So the first, and that gives you an idea whether this organism you can play around with, and what are the targets, and so on and so forth. So essentially we start with, of course, and there are many different kinds of tools for this flux analysis. I'm not going to get into that, right? So one can do this and then predict what's going to be the ideal pathway, and what are the competing pathways which should be knocked off, and if the competing pathways are knocked off or down-regulated, what's going to be the effect on the organisms, and so on, right? Also, in the pathway of interest, one can go about, engineering the enzymes itself. So the enzyme design will also plays a crucial role in metabolic engineering. You can either look at the activity of the enzyme, whether that should be produced in high amounts, what is its catalytic activity. You can look at whether it's feedback regulated, whether you should remove the feedback regulation, okay, or whether in the regulatory sequence for that enzyme production, are there genetic regulatory elements that one should tinker with, such that the enzyme is expressed in optimal amounts. And all these things, of course, one can use using standard tools for genetic engineering. Okay, so I'm not going to get into all of these things, and whether you play around with just the gene expression strategies, playing around with the promoters or the ribosome binding sites, etc., or you play around with the secondary structures of the RNAs, and this is something I'll talk about in detail later, whether you want to regulate at the translational level, etc. There are a lot of these tools, and I think many of the previous speakers have talked about these tools, so I'm not going to look at these things in much detail now. I'm going to talk about a couple of things about promoter engineering and about RNA-based regulation, but trust me, there are a variety of other tools. Of course, all of you would have heard about CRISPR-based tools for gene editing, which can be used also not only for knocking out genes, but also CRISPR-based interference, which can help in down-regulating the expression, down-regulating the flux through a pathway, or down-regulating certain genes, right? One can also use tools to combine the enzymes in a particular pathway and localize them on a particular scaffold, such that the intermediates in the pathway do not get diffused in the whole organisms, but if there is a localized high concentration, they can tunnel through the pathway, and that can happen if all the enzymes are in proximity to each other. So that can be done, or sometimes the enzymes can be placed in a micro compartment, if that is where a lot of the bioconversion would take place, be it in a mitochondria or a lysosome or some vacuole, etc. So, or it could be membranes, right? So all these things, are ways to go about engineering the organism. It's not just often enough to overexpress the genes or just express a heterogeneous set of genes and you think you're done, right? There are also sometimes a particular organism where if you want to produce a particular product, it may involve engineering a very long pathway. And that may not work too well, especially if there are a lot of regulatory constraints. So sometimes one can, use multiple organisms in the same culture, what we call as co-culture, right? So the end product of a particular pathway can be taken up by a second organism which is engineered, and that second organism could produce the product of interest. This is something we have done in our laboratory, right? We had a particular organism which took up lactose, and we wanted to convert that into D-lactic acid. D-lactic acid is an isomer of the common lactate, L-lactic acid, and it's much more expensive and it has a lot of applications in bioplastics. Okay, so but what we found is that the lactose which was being taken up, only the glucose part of it was being used to produce D-lactate, and the galactose was not used, and so galactose was being accumulated in the culture. So we went and engineered another lactic acid bacteria and made it take up only galactose and convert the galactose to D-lactate, right? So that can be done. Or sometimes people can make organisms convert glucose to pyruvate very efficiently, spit out pyruvate, and that pyruvate, which is an important intermediate after the glycolytic pathway, can be used for a range of different products. So one can actually have co-culture, or one can have co-culture where an organism breaks down toxic metabolites, which is there in the fermentation media, or toxic metabolites which are produced along with the main product as a byproduct, and so on. So there are a lot of different things one can use metabolic engineering for. So these things can be done. And then, of course, there are what we call as evolutionary engineering strategies and tools. And this is the beauty. Often, you know, we can do very rational metabolic engineering, very directed metabolic engineering, but that may not give us the results we want, right? Over the last 100 years, we have known that organisms, if they are randomly mutated and screened, sometimes, give us very unexpected results, give us much better productivity, right? So there is another technique called adaptive laboratory evolution, whereby you subject organisms to different kinds of selection pressures, be it growing on different substrates or on a high substrate concentration or withstanding high product concentration and so on. So what happens is that you grow the organism in these extreme conditions and slowly, well, not suddenly, but slowly increase the condition under which an organism can adapt to an extreme condition. And then what happens is that at a particular condition, the organism accumulates a lot of genetic changes. It is able to do things much more efficiently than what even your directed engineering can do. And that is because, it's just evolution. And the organism accumulates changes in certain genetic, there are certain genetic changes which you will not even start to predict, right? And that's what I mean. We don't understand biology enough. And therefore, such organisms, if they are much better, then you can sequence them and then find out where the genetic changes have occurred and do a reverse engineering, right? So we have done some of these things in our lab, whereby an organism is made to grow by evolutionary adaptation at much higher temperature or much lower pH, and then you use that organism for metabolic engineering. So these are strategies. But all these things, as I said, will not happen at one go. You cannot do directed metabolic engineering, you cannot do protein engineering, design a lot of different pathways, knock out a pathway here, knock out a pathway there, express multiple genes, and and expect things to work. It never works that way, right? It's a very iterative process. And I think people would have talked about this, or you might know about it, what we call as the build, test, learn, design cycle, or the design, build, test, learn, design cycle. So essentially, we start with an organism which is either well known, and we use the computational tools to design pathways and knock out other pathways and so on and so forth. So in this process, you have a tentative idea that if I do metabolic engineering, and this is all computational, right? Then it will work in a certain fashion. And then you go about, from that computational models, you go about knocking off specific genes and pathways. You go about go about overexpressing genes in certain pathways, and so on and so forth. Or you may use intuitive knowledge from fermentation, right? And from there, you may know that, okay, certain byproducts are being built up, and how do you go about reducing that byproduct formation, and so on and so forth, right? So, or sometimes you may have an organism which you don't know at all. Normally, all this is done with well-known organisms, E. coli or Saccharomyces, etc. But what happens if you don't have a well-known organisms? Then it becomes even more problematic, because you'll have to develop what we call as a bacterial chassis, or an organism for metabolic engineering. So you'll have to start obviously with genome sequencing tools, know the entire genome sequence. You'll have to get all the omics data while this organism grows, find out what genes are expressed, find out the fluxes in different metabolic pathways, and so on. And then, you'll know from these you can build metabolic models, and then one goes about validating these metabolic models. So you need to have tools. So along with all this data that you generate, you might be able to generate a range of tools. And I'll talk about this in the synthetic biology part in more detail, where you have promoters and plasmids, etc., and be able to apply all these genome engineering tools or genetic engineering tools for engineering of the pathways, right? You would also perhaps want to create a range of different organisms, or what we call as a mutant collection. Nowadays, this is easily available for E. coli or something like that. There's a library, knockout library available. But what if it is not, right? You might want to create a knockout library. Now it becomes much more easier with CRISPR and so on and so forth. Earlier, it wasn't so easy, right, to have a directed knockouts and create this library. Or you might want to create what we call as a reduced or a minimal genome, whereby the unwanted genetic parts or genes are all taken out and you work with a minimal genome. And remember, all bacteria evolved to withstand a variety of different conditions. But in a metabolic engineering case, in a bioreactor, you work only in a narrow defined range of conditions. So the bacteria does not need many of these genes for adaptation. So we can create what we call as a reduced genome or a minimal genome, right? So this is how one goes about designing an organism, and then you can build all the pathways, etc., and engineer the organism, right? After you build, I think the important next step is to test or validate whether this organisms will behave the way you have predicted it to behave theoretically in your simulations, okay, or even intuitively the way you have predicted. Does it give similar kinds of results across shake flask, laboratory scale bioreactors, pilot scale bioreactors, and large scale bioreactors? If not, if it doesn't, then there is a problem, right? You take the data, you go back to the drawing board and redesign. So you keep on following this iterative loop, right? All this data which is taken is stored in the learn phase and can be used to improve the design. So you keep doing this until you get to a point where the organism is meeting your objectives. You can set what are the objectives. I want 100 grams per liter of this particular product. I want production of 1 gram per liter per hour productivity, and I want maybe 60% of my substrate converted to product, and so on and so forth. So you may not, of course, get everything, right? There are always trade-offs between these criteria, and that's one thing about try and talk about. But if you get some reasonable results, then you can say, okay, this is fine enough, I'll go ahead. And this comes close enough to what is an industrially defined criteria, and this will be economical and so on. But this takes a very long time, right? And that is why, I didn't show the slides, but most organisms to engineer take 20, 30 man-years to build, right? Often maybe 5 or 10 PhD students working at the same time or in series, and over generations to build. And with the papers on it, anywhere close from 25 to $50 million to build an industrial scale organism. You might think this is very expensive, right? Why should I get to that, and who will invest in all that? But once you do it, it becomes very cheap. Once you do it for an organism, that organism can be used for engineering many other processes and products. Once you do for that particular organism and you get the knowledge, that knowledge can be applied to many other organisms, right? But somebody has to bite the bullet and initially do this. And that is what many of us metabolic engineers work on for generations together to get to the point whereby the built-in knowledge can be used by many people. Okay, so I'll start to get into synthetic biology. All this metabolic engineering, how do we go about to do it? Can we use the kind of approaches which are used in traditional engineering to understand and build biological systems? And that's where synthetic biology has come, right? Synthetic biology is a much more a fancy which was caught up by physicists first. Just like in molecular biology, physicists played a huge role in the revolution of molecular biology, right? Big people like Francis Crick or Max Delbruck and so on and so forth. Many of these people, came from asking questions which are generally asked in physics and engineering and saying that, can I apply these kinds of thought processes or these kinds of framework in biology? What are these questions that physicists ask? What is this framework? And these are things that are done in mostly electrical, mechanical engineering, and so on and so forth. You know how all these parts function, right? Whether it's a capacitor or a resistor or a pump or some valve, right? Or in an automotive car, if you apply a brake, you know that you can stop a car from 60 kilometers per hour to zero within such a distance. You know how this is function, and whatever you have designed, you get the output according to the design process, right? In biology, it doesn't work that way. So in most other engineering, parts can be constructed to function according to design. If it didn't happen, right, all your computers won't function very well, all your cars and aircraft, etc. You cannot predict it. You will have far more failure, right? Every time you take an aircraft, you fly, you are confident it's going to fly, it's going to land, right? There are millions and millions of flights where there is no problem. And all of these follow the Six Sigma rule of quality. But in biology, that doesn't happen, right? So the question is, when all these parts are assembled, will they behave according to design, right? Right from the most basic set of components, right, which are used in electronics. The capacitors and transistors, etc. They're combined to logic gates. These logic gates can be combined into integrated circuits, and you can build a lot of devices, computers, and so on and so forth. And right from the most basic physical layer, you can quantitate the value of a capacitor or a resistor, how many ohms, micro ohms, or how many microfarads, and so on and so forth. You can look at the input-output function of a logic gate, right, of a transistor, and it will exactly behave according to simulation. You try to do that for biological systems, it doesn't work, right? You put together a genetic network which will, have a number of genes built together into a metabolic pathway, and those pathways, different pathways can be constructed into a cell, and you say, okay, I'm going to give you so much amount of input, so many grams of glucose to be converted to so much amount of product in 2 hours. And this is what my simulation says. Are you going to get the result? No, right? So that's a challenge in biological engineering. It's going from defining the molecular components to stitching together the molecular components which have certain values. You may know that a particular enzyme has a particular catalytic activity, right? You may know that a gene, when expressed under a certain promoter, may produce certain amount of protein in certain amount of time. But if you do all this, will the organism finally give you the output that a particular enzyme does, or a particular set of enzymes? Will it give you a particular output in terms of flux through pathway? That is not there, right? We are still not there, and that's the problem. And why is that so? There are many issues, and I'll kind of quickly go through some of these. But each of these can take a whole lecture, right? I talked about the fact that most of the parts in biology are unknown, be it genetic parts, be it how the genes are regulated, be it what is the function of proteins, be it how different proteins interact with each other, what are the different transcription factors which come together to regulate gene expression, what are the things which can inhibit gene expression, what are the repressors, how would a particular repressor interact with other parts, metabolites, etc., to repress gene function. Many things are known, but across different metabolic networks, across different sets of genes, many things are unknown, right? So qualitatively also we don't know. It's like saying we know all the actors in a movie, but we don't know what roles they play, right? We don't know which actor loves which other actor, who is a villain, who is a hero, we don't know. We only know, the different actors. Then we also don't know the parametric values of many processes within the cell, right? The rate of transcription, right? The rate of catalysis, okay? The rate of transport of something across a cell, and so on and so forth, right? So even if we qualitatively know how a gene is triggered and what are the regulatory bottlenecks, and what is the repressor and what repressor binds, we may not know what is the strength of the binding of the repressor to a particular regulatory part. We don't know the transcription rate. We don't know the rate of protein folding, and so on, right? So it's very difficult to predict how things are going to work. We also don't know what are all the interferences from the organism onto a particular part or a particular genetic circuit that you have engineered, right? So we don't know whether a particular transcription factor which works for the host pathway is interfering in the circuit that we have engineered, or whether the RNA polymerase, how it will interact with the regulatory components in the heterologous circuit, and so on. We don't know, once we start expressing many of these genes and proteins, how the cell will interact, what we call as the metabolic burden, how the cell will share resources, and when it's starting to share the resources, such as the amino acids for making a protein or the ribosomes for translation and so on and so forth, how does the sharing takes place? Would the cell start to shut down the, some of the machinery within the cell? Would the translation machinery be down-regulated, and so on and so forth? With many of the metabolic pathways, the catabolic pathways especially, how that would get down-regulated if you're going to invest a lot of resource into producing certain proteins. We don't know this, right? And how do you go about controlling? We don't know the heterogeneity in a cell, right? There is a lot of this so-called noise, right, which can lead to a lot of this heterogeneity, and that you see in a microbial population here, from green to orange to red, which can lead to cells having different ages, right, different levels of gene expression. If all the gene expression were homogeneous, you would get pretty much all cells grow in synchrony. But often you get a heterogeneity of gene expression. And so you might have some cells expressing the genes in a high amount or having a high plasmid copy number, some cells having a low plasmid copy number, and so on and so forth. So there are all kinds of intrinsic noise within the cell which can create this heterogeneity. There's a lot of extrinsic noise, how cells react to different environmental perturbations, be temperature, pH signaling, and so on and so forth. Okay, so all these things, uncertainties, need to be overcome right? So we need to first have a comprehensive quantification of many of the parts, and that is being done in a lot of the modern biology, although it's very, very expensive, but it's been happening over the last 20-25 years. You know all this, I'll not get into details, right? All the omics, not just the genomes, the RNA, the transcriptome, the proteome, and so on and so forth. But all this is getting extremely expensive. So unless a lab is very well equipped, or unless the consumable costs are really low, we cannot really do this. That's a problem. But tomorrow, all these things might get integrated into a lab on a chip, and you may be able to do much more cheaply. Today, the cost of genome sequencing in just 20 years, the cost of genome sequencing has fallen by an order of 6 orders of magnitude, right? Where it used to cost $1 billion to sequence a human genome 20 years ago, just 20 years ago. Now it costs on the order of a few $1,000s. So that's an amazing thing. And that might happen to all of these things. And once it happens, it will be very easy to characterize an organism, and not just reading a DNA, but writing a DNA also, those costs need to form. And so today, the kind of molecular biology that you do in terms of restriction, digestion, ligation, putting things together, in 10-15 years, that will be history. Nobody will be talking about restriction, digestion, and so on and so forth. You will just synthesize a plasmid, synthesize an organism from A to Z, right? From knowing what the sequence you want, you can synthesize a plasmid. That's already being done. You can outsource it and do it today very easily. But synthesizing an organism is tougher. But give it another 5-10 years. Excuse me. Sorry. Give it another 5-10 years. It will be able to do it. So this is completely going to revolutionize molecular biology. So all of molecular biology tomorrow is going to be just you sequence, you tell what you want, you build parts together. And I'll come to that synthetic part, right? And you just plug, play, and go. Well, not that simple, but we are getting there. Right? We also want to optimize parts and do all these things. So, okay, I'll not talk about all this building regulatory circuits which reduces noise, so on and so forth, but you get the idea. So for this, you need the kind of toolboxes for biological engineering, which doesn't exist to the same level that it exists in other engineering sciences, right? In all other engineering, electronic engineering or aircraft design or chemical engineering, etc., you have standard tools for testing, for measurement, right? You have oscilloscopes, stress meters, you have pressure gauges. You have all kinds of toolboxes. You don't have even toolboxes for measurement of biological process, although now you have it, right? With fluorescent proteins and so on and so forth, and you can easily do qPCR and so on. Now you have toolboxes for modeling and simulation of biological phenomena, right? You can easily get standard software for metabolic flux analysis, for any kind of simulations, right? So you need to generate many of such toolboxes. The question is, does biological engineering have its own toolbox? When I said toolbox, it's not simply a matter of saying, oh, okay, I can easily buy restriction enzymes, and I can get plasmids, and I can engineer all these things. That is fine, but that's old hat. If somebody is going to build a car from scratch today, none of the car makers do it. I mean, they'll laugh at you. If you are going to say, okay, I'm going to make chassis, I'm going to make wheels, I'm going to make, all other parts. People just don't do that anymore. They just assemble, right? So tomorrow in biology, when we talk of a toolbox, it means all these tools are available. You just get all the parts and assemble it. And so even a car, when it is assembled, the parts come from 20 different countries, where all these countries where people are making these standardized parts which will fit in. Every nut and bolt would have a very precise, design, a precise size, precise screw it will go into, and so on. If it doesn't, it's gone. The car cannot be assembled, right? If it's an electronic circuit, if all these parts do not have precise values, and if you solder, you will not get the result. So in biology, we are moving towards making such toolboxes. What does that require? That requires standardized parts, and that's available. It started with this whole competition called IGEM. You would have heard about it. It's called the International Genetically Engineered Machines Competition, right? Where many of these standard biological parts are there in a repository. And even that's going to become old hat very fast. Right now, it's in a repository where people say that, okay, please send me this DNA, which does this function of a promoter or a reporter protein and so on. And they will ship it to you in a standard plate. Tomorrow, you will just go to that library, know what that sequence is, that promoter sequence which reacts to particular condition, right? What is the sequence which is a codon optimized reporter protein for Lactococcus lactis? That will be there. Then you will tell some company, or you will have it in your lab, you will just put the oligonucleotide, synthesize that part, and be done. So even that repository will not become a physical repository. Right now it is. Right now you can see all this in the standard registry of biological parts. You have this catalog, right? Composite parts, and then people they'll tell you what collections we have and how it is documented and so on. And then if you ask them, they'll tell you protocols, how to put it together and so on and so forth. Tomorrow, this physical library is gone, right? And in a sense, that's happening in manufacturing, right? You can have a 3D printer and you know how something should be put together. You can easily design a part to what you want. You don't need to buy it. Of course, there is material cost and it is easier to do it. It's easier to outsource. But I'm saying that how this biology revolution is going to change the way we are going to do biology. So this is all there. For example, you go in this registry of biological parts, you go to this, whatever documented parts are there, go to this collection and say, okay, I want to see what are all the different promoters, a promoter catalog, right? It will give you a certain references. They'll have a part number to that promoter, what we call as a biobrick library, and that parts will be defined, right? And if you go to that part and find out, okay, in that catalog, right, how to work with this promoter, there'll be protocols which will tell you. It will tell you what the promoter tells, and that it will tell you protocols of what promoters it has worked with, right? So there will be parts which are extremely well documented, and there'll be composite parts. There'll be promoters which is put together with some other regulatory part, okay, which does something, and you can take all these things and use that composite part in your device. So that is for synthetic biology. I'm not going to get into all that. And all these things, parts, required certain properties. It's not just enough to build this library. What are these kinds of properties we are talking about? Right? And it takes a lot of work to define the properties of these parts. What are the desirable properties? So in these synthetic parts, to make things work as we put together, they need very different kinds of properties. They need properties like independence. That one, it does not interfere with the host cell machinery. Okay, I'll not talk about all these parts in detail, but I'll give you some examples. So it requires independence, tunability, orthogonality, scalability, and so on. And I'll just describe this in a little more detail. When we talk of independent parts, it means that these parts do not interfere with their host circuitry and vice versa, right? They don't interfere with each other. For example, if a repressor binds to a particular promoter, it will not bind to other promoters. If a particular, RNA polymerase binds along with a sigma factor to regulate the gene expression, these two do not do that for other parts. So that's important. And then there are fact that you should be able to take this part and put them together in a modular fashion across different genetic circuits. The same promoter should work in expressing different sets of genes, right? If you put it in a particular organism, it should under a particular environment be able to give you the same kind of expression. That's what we talk about, robustness and reliability and so on. Tunability is important for controlling gene expression, right? It makes, and so one can do that by varying the promoter strength, right? I mean, gene expression, of course, depends upon a variety of different conditions, right? The growth of the organism, what is the temperature, pH, and so on and so forth. And so how do you measure the strength of a promoter? So one needs some kind of a standard promoter against which we can say that this is the promoter strength. Just like how do you know that something is 1 meter long? That's because there is actually a part where a particular length defines a meter, and according to that length, everything is defined as 3 meters or 3,000 kilometers and so on and so forth, right? So when you talk of a promoter strength, you need to have a standard part, and that can be done by having the standard promoter in a plasmid and another unknown promoter in a plasmid, expressing two different fluorescent proteins. And one can measure the promoter strength with respect to the standard promoter. Okay, and that can be done across a variety of growth conditions, and people have actually done that, right? And they have looked at the characteristics of different promoters, the characteristics of the control promoter, and this is the characteristics of the unknown promoter. And as you can see, across a range of conditions, these have the same slopes, which means that this ratio works very well. And using that, one can build promoter libraries. I'll not get into details of this, but that allows us to have tunable promoters, and the fact that if you can have tunable promoters, you can control gene expression across different metabolic pathways or metabolic circuits. Orthogonality is important, right? Which means, can you separate the gene circuit that you're trying to express, or that you're trying to, or device that you're trying to express in an organism from the other parts of the organism? Can there be non-interference, right? So orthogonality has an overlap, what we call as independence also. But in this case, I'll give you an example. One of the best example of orthogonality is the T7 promoter, right? The T7 promoter works with the T7 RNA polymerase, which will not express the other promoters in the organism. This T7 RNA polymerase will only bind to a T7 promoter, and so it has great advantages. It is completely orthogonal to the organism. So you can control the induction mechanism and so on and so forth. And people have used this property to build range of pairs of T7 RNA polymerase and promoter, what they call as cognate pairs, which don't interfere with each other. So you can see here that this particular mutant of T7 RNA polymerase will work with this particular T7 promoter, and there can be another mutant which works with another promoter. And as you can see, there's a lot of orthogonality. This particular pair of T7 promoter and T7 RNA polymerase will light up very well only when both are there. It will not light up too well when it is a different promoter or different polymerase. So you can have a range of polymerases, and you can see this, right? And this is a logarithmic chart which shows that a particular promoter polymerase pair can be completely orthogonal. And so therefore, you can use this across different genes within a circuit, and the T7 RNA polymerase can be triggered by different kinds of input functions, not just the IPTG alone. Okay, so by that you can switch on, switch off different sets of genes which are controlled by different T7 promoters, which respond to different T7 RNA polymerases, and therefore you can have much better control over a genetic circuit. right? So that kind of orthogonality can be done with promoters, but it has also been done with the ribosome binding sites and 16S RNA, right? People have worked with mutant ribosomes that recognize very specific kind of 16S RNA sequences. So that is the beauty of synthetic biology. And of course, composability means that you should be able to put all these parts together in a way that all these things work, will be it promoters or ribosome binding sites or reporter proteins or terminators and so on and so forth. And the fact that if you are able to compose it, this should work across different genetic circuits, across different organisms. So scalability means that all these parts that you have put together are not only independent and robust and orthogonal and composable, but can be put together in variety of ways, which leads to different genetic circuits or metabolic pathways and so on and so forth. So that's the kind of parts that we're talking about when we build libraries, right? When we talk about these desirable properties, when we build libraries of different parts, this is the kind of standard biological parts libraries we are taking. Of course, it takes a lot of work, but once it is done, once there is reliability, right? You don't need to build a physical library. All you need to do is have a library where a sequence is there. You can just patent it and work with it. So once this is done, right? Once this is all that is done, then this is where synthetic biology begins. So essentially, you need all the knowledge of the sequence data, etc., hierarchy of how things function, standards, and then you need to put all this knowledge together into what we call as tools for fabrication. And today is just cloning and restriction, ligation, etc., and finding ways to put parts together. Tomorrow it will be simple DNA synthesis, right? And how do you put all these parts together for useful purposes? So at this point, I'm going to switch gears. I know I'm running out of time, so you can always interrupt me if you say if you have had enough and say that, okay, we don't want much more talk about synthetic biology. So you can do all these things for controlling many parts functions. And why do we want to do this for hyaluronic acid? Essentially, the idea here is that, I know I'm getting questions, but maybe I'll answer that at the end of it, another 10 minutes. The idea is here, if we want to control, are we just happy with producing hyaluronic acid? Do we want hyaluronic acid with a certain titer and productivity rate, or do we want certain things that we want to control like molecular weight? Remember, I talked about that earlier. And that's been the goal in our laboratory, right? Why do we want to do all this? Hyaluronic acid has a huge commercial market, right? The global market, more than $10 billion by now, right? It is used for a lot of different applications, as you can see here. Main applications, of course, are for things like arthrology or, osteoarthritis, by which it can be given as injections to find joint pain, or it can be used in ophthalmologic purposes in eye surgeries, or it can be used in wound treatment or drug delivery. It can be used as cosmetics, right? In fact, the bulk of the applications go into cosmetics, or it can be used into tissue engineering or tissue regeneration. So a lot of applications are there, some of which like cosmetics and arthritis are the major applications, but there are investigations like for other biomedical polymers and using HA for a range of applications, right? And so all these applications require a lot of range of different molecular weights. As you can see, for viscosupplementation for arthritis, you may require very high molecular weight. And as you can see, this is obtained by cross-linking HA, which is obtained from rooster comb or from bacterial source. So I need to do metabolic engineering just to not extract from rooster comb. We're all now wary of animal products, right? COVID has told us, stay away from using animal products for a lot of different reasons. But I also need to control molecular weight. Question is, can I control molecular weight? by metabolic engineering alone? Yes and no. I need to use a variety of different tools. So I'll get away from this. I'm just saying that metabolic engineering for molecular weight control is very, very important. So we have done a lot of different things in our lab for enhancement or control of molecular weight. I mean, we started very simply by looking at overexpression of multiple genes, and we can see that by using a combination of genes, either using this Has A and Has B alone, or using A, B, and E, or maybe using A, B, and C, or A, B, and M, or A, B, and D, which can get not only hyaluronic acid of different concentrations, and of course all these concentrations depends also on the amount of glucose that is used in the shake flask. So it also depends on environmental factors, but that you can get different molecular weights, right? Just using A and B gives you a low molecular weight. Using A and B and E gives you much higher molecular weight. Using 10 gram per liter glucose gives you slightly lower molecular weight, but if you use the same construct, much higher glucose concentration, it gives you higher molecular weight. So all these combinations are there, right? And in all this, you would have seen something else. I'll just point it out, that there is a molecular variation within the experiment. There is a peak molecular weight that one gets, but at the end of the fermentation, the molecular weight is much lower, and that is because the enzyme here responds to the molecular weight. The enzyme here, hyaluronan synthase, responds to glucose concentration differently and will start producing shorter chains during the course of the fermentation. So one has to devise process strategies for molecular weight control of HA, and by using glucostat strategies by which we maintain glucose concentration constant. We'll not get into those process in detail, but as you can see, if we maintain glucose concentration constant through the course of a process, and all this we have done, we can maintain the molecular weight constant, right? But in all this, we can vary the, we can maintain constant molecular weight at different values, from 0.4 million Dalton to maybe around 1.4 million Dalton. So you can get 0.4, 0.8, 1.2, 1.4, and so on with different constructs. So the idea is, in all this, to cut a long story short, we need to control the pathway fluxes, right? In these two major pathways which produce the precursors for the two precursors for hyaluronic acid. So the ratio of the fluxes in these two pathways will determine the ratio of concentration of these two precursors, that is glucuronic acid and N-acetyl glucosamine. And as you can see, the ratio of these fluxes, and the same thing holds true for precursor concentrations, can give rise to different molecular weights. So as you go towards a more balanced ratio of fluxes to a value of around 1 between these two metabolic fluxes, you can get to a much higher molecular weight. But what if I want to do a different molecular weight? Maybe I need to have unbalanced fluxes. So all these things can be done by controlling both, not only the ratio of fluxes, but how much of these glucose is fluxed into the catabolic pathways. That will also control molecular weight. So essentially, what the tools we want to employ, and I think I will stop here because it's already getting to 1:30, and because talking about riboregulators will take another 15-20 minutes, but I can come back to the question answer session. The tools we want to employ would control the flow of glucose not only into the catabolic pathways, and these are the major competition pathways, but we cannot simply knock out the glycolytic pathway or the pentose phosphate pathway, right? We can perhaps regulate the flows into these pathways by having what we call as riboregulators, which control the flux through this pathway, or riboregulators which control the distribution of fluxes through these two pathways, which will allow us to control the final molecular weight of hyaluronic acid. That is what we want to do. That is what two of my students have been doing in building this riboregulator platform. And what is that? Just one more slide. And essentially, these riboregulators are used for control of gene expression. It involves a small sequence, what we call as a cis-repressive sequence, which is engineered upstream of the gene that we want to express, of the protein we want to make. And when we express the RNA, the cis-repressive sequence sequesters the ribosome binding site by forming a secondary structure. Okay, so the mRNA will form a secondary structure, sequester the ribosome binding site, and not allow the translation of this mRNA to this particular protein. In this case, we are talking about reporter protein. But if I activate another RNA called the trans-activating RNA, and that can be done independently, then this trans-activating RNA will then go and destabilize this particular secondary structure, and it will destabilize by initially binding to this loop here, and it will destabilize that loop and then form another secondary structure. So this part of the cis-repressive sequence which you see in the orange will form a stem loop structure here with the trans-activating RNA, which has a complementary sequence to both this gray part here, which is the loop, and this orange part here. And then it will form a secondary structure here and thus free up the ribosome binding site, which will allow the translation to occur. And therefore, this will allow the gene to be expressed. Right? So this can have a lot of advantages in terms of tunability, in terms of independent regulation of multiple genes, in terms of fast response time, all this desirability that we talked about in metabolic engineering. So it can have, and you can control the expression of many of these different genes, not only in the pathway of interest, but in the competing pathways. And that is what we want to do. And get to a point where we can control all these metabolic pathways in a tunable fashion so that one can get the final output, which is desirable molecular weight, or perhaps sometimes it could be desired, titer value and so on and so forth in the product of interest. So at 1:30, I'm sure many of you will be hungry and tired. I'm going to stop here. Let me see how many slides were left. I'll just go to the last slide. Yeah, there are only about, oh wow, actually there are about 10 slides left, so I'll not get into all that. I just want to acknowledge many of the people who have contributed to all this work over the last 8 or 10 years, right? Many of them have finished their PhD and graduated, at least 4 of them from my lab. Dr. Shashi, Dr. Ashish, Dr. Srija, Kirbakran, Dr. Andabharti is a postdoc who's stitched together a lot of the groups and variably contributed. Many of them are just graduating. Dr. Pandishwari, who worked on the, many of the glucostat process and building the library. Sandeep and Richa, they're not here. Sandeep is here. And perhaps Richa, where is she? Yeah, she's here. So they are making these riboregulators in the library. So as I told you, this takes enormous amount of work and many number of projects and many tens of student PhD years to start to get to a point where you can have a synthetic biology application for metabolic engineering. Okay, so all this has just been done in Lactococcus lactis. We are now doing this with some of the other strains for other applications. So just to tell you where we are and what we are doing, may seem a small amount of work, but trust me, it takes a lot of money and effort. Okay, so I'll end here. If you have any questions, I can go back and ask about riboregulators or anything else. I'm happy to answer. Sorry, get to the
It's a really great and fantastic, Professor Jayaraman, that you went length and breadth of the metabolic engineering to, present the revealing stories about foundations, fundamentals, and classical example of hyaluronic acid production by using alternative microbes and other things. It's really fascinating, sir. And so I thank you at the outset.